I'm Donald Lloyd-Jones from Boston University School of Medicine, PI of the Framingham Heart Study, and a past president of the American Heart Association. We're coming to you from New Orleans at the AHA Epi Lifestyle Conference for 2025. And I'm so delighted to be joined this morning by Tess Peterson, who is the winner of this year's Jeremiah and Rose Stamler Young Researcher Award. Now, for those of you who don't know, Jerry and Rose Stamler actually founded the Department of Preventive Medicine at Northwestern, and they were leading lights in defining the field of cardiovascular epidemiology. So Tess, congratulations on the win. Really, really wonderful science. Tess's science addressed gene expression profiles in circulating monocytes and how they relate to cardiac remodeling and incident heart failure from the MESA study. So Tess, if you would tell us a little bit about your research.
Absolutely. First, I just want to say what an absolute honor it is to be here. Very big deal for me, so thank you. So inflammation's been linked to heart failure for decades. So much so, in fact, that clinical trials have aimed to target inflammation in heart failure for a long time, and they've largely yielded disappointing results that have taught us that if we want to target the immune system, we need to do it in a more nuanced way. And so this work specifically focused on monocytes, which are a type of immune cell that are integral to inflammation and tissue remodeling that we know is happening under, cardiac stress that can ultimately lead to adverse cardiac remodeling and incident heart failure. But we really don't know a lot about the role of circulating monocytes in this domain. So the objective of this study was to generate hypotheses on specific monocyte-mediated biology that could potentially contribute to adverse cardiac remodeling and incident heart failure.
Perfect. So you pointed out really nicely in your wonderful presentation, we think of monocytes as always being bad.
Yeah.
But there are some really helpful aspects of monocyte activity that lead to healthy healing of damaged myocardium and things like that. Could you talk a little bit more about that?
Absolutely. So monocytes are really interesting cells. They are highly heterogeneous and highly plastic. So they're constantly adjusting their gene expression profiles to adapt to what they're needed for. And so they could serve functions that are pro-inflammatory or pro-resolving. And really, we've learned in the last, decade or so that that really is a functional spectrum that they're moving across continually. And so they might be contributing to tissue remodeling specifically by phagocytosing tissue debris or secreting effectors that could stimulate extracellular matrix remodeling and cell proliferation, angiogenesis. So they could be contributing to remodeling in a wide variety of ways, and we don't really know a lot about that balancing act. And so this was really phenomenal data that I had a privilege to work with to kind of we really couldn't disentangle a lot, but to start generating some hypotheses on a population level.
Yeah. Tell us a little bit more about the specific findings that you were able to garner in MESA.
Yeah, absolutely. The study was conducted in MESA, which we're all familiar with. And in a subset of MESA participants that we isolated circulating monocytes and measured gene expression profiles within those monocytes and related them to various measures of cardiac structure and function ascertained by gold standard cardiac MRI. And so we evaluated 6 cardiac MRI characteristics, and those are cross-sectional analyses, and in those analyses we identified profiles that were suggestive of, were enriched really for biological processes related to T cell and B cell activation, cytokine production, extracellular matrix remodeling, things related to tissue homeostasis. So a wide variety, phagocytosis, apoptosis, I'm forgetting a few things. So really functions that span the spectrum of monocyte activity. So very, very interesting that we're seeing things across that functional spectrum there. And then we sort of used that as a bit of a filtering step and ultimately identified 96 transcripts that were associated with more than one adverse cardiac characteristic, which we then evaluated for associations with time to incident heart failure. And we identified 3 genes whose monocyte-specific expression was associated with heart failure. And those genes serve roles that are, may biologically
Plausible.
plausibly,
Yeah.
contribute to disease. And there's murine model data that's even suggestive of it. So very exciting and very, very cool findings. And I'm excited for the next step.
Well, terrific work. And you anticipated my next question. What are the next steps?
Yeah, so I think, the natural next step has to be validation, right? And that could take several different forms. One approach could be functional studies, moving to a mouse model. Obviously, it would be fantastic to generate similar data in an external population. That's, yeah, that
Yep.
would be
Yeah.
fantastic.
And ultimately, maybe these three genes, in an ideal world, could potentially be targets to interrupt some of the processes that lead to heart failure.
That's right. I personally believe that, the immune system is so complex. We can't be targeting sort of broadly active immune mediators. And so if we could, sort of, uh, take advantage of that complexity and target more specific immune mechanisms, there's potential there.
Yeah, potential precision medicine in action for the
Absolutely.
future. Wonderful. Now, Tess, you shared with me that you are a freshly minted assistant professor at
That's
Johns
right.
Hopkins. Congratulations.
At Minnesota, University of
Oh,
Minnesota.
my apologies.
Transitioning,
University
yes.
of Minnesota,
Yes.
transitioning from your postdoc at Hopkins.
Correct.
Wonderful.
Yes.
Thank you. Any advice for our young investigators out there who are postdocs or newly appointed as young faculty? What have you learned in this journey?
I think, the biggest thing for me that has led to, the success I feel I'm having right now is my mentors. Find mentors that can really advocate for you, and they're out there. There's plenty of them in this community, that's for sure. And so I would certainly not be where I am today without my fantastic mentors.
Terrific advice. And mentors are busy people. How do you manage your
Yeah.
mentor?
If you find the right mentor, they make time for you.
Yeah.
I had weekly meetings with my mentors, and if that was on Zoom, that was fine.
Yeah.
Just touching base and talking about progress and, keeping that connection moving and alive.
Wonderful advice.
Yeah.
Terrific. Well, Tess, again, congratulations on all your success. We look forward to your future research endeavors, which I'm sure will be equally successful.
Thank you so much.
And congratulations.
It's an honor.